Welcome to this lecture on Reptilia classification. Today, we are going to explore how ancient creatures conquered the land and how scientists use the architecture of their skulls to understand their evolutionary journey. It is a fascinating story of survival, adaptation, and biological engineering. By the end of this session, you will see these animals in a completely new light. So let us dive right in. To understand reptiles, imagine designing a vehicle that must leave the water and survive in a scorching, dry desert. You would need tough, waterproof armor. This is exactly what nature did when amphibians evolved into reptiles. They were the first true land conquerors. By developing waterproof, scaly skin, they prevented their bodies from drying out under the harsh sun. This crucial adaptation broke their final tie to the water, allowing them to explore and dominate entirely new terrestrial ecosystems that amphibians simply could not survive in. Now that we understand how adult reptiles survived on land, let us look at how they protected their young. The secret is the amniotic egg. Think of it as a portable pond. Before reptiles, animals like frogs had to return to the water to lay their jelly-like eggs, or the eggs would dry out and die. The amniotic egg changed everything. It contains specialized membranes, such as the amnion and chorion, which protect and nourish the embryo inside a safe, fluid-filled environment. This incredible evolutionary leap allowed reptiles to reproduce anywhere on land. Building on these evolutionary leaps, how do we formally define the class Reptilia in biology? They are tetrapod vertebrates, meaning they have 4 limbs, and they are ectothermic. Ectothermic means they cannot generate their own internal body heat. Instead, they rely entirely on the external environment, like basking in the sun, to regulate their temperature. Furthermore, their bodies are covered in tough epidermal scales made of a protein called beta-keratin. This beta-keratin layer is essential because it acts as a barrier, preventing desiccation or water loss while providing excellent physical protection. So, which animals actually belong to this group? Traditionally, the class Reptilia includes familiar faces like turtles, crocodilians, snakes, lizards, and the rare tuatara. However, modern science has given us a plot twist. Modern cladistics, which groups organisms by common ancestry, actually places birds within the reptilian clade. This is because birds and modern reptiles share a direct dinosaurian ancestry. Despite this genetic reality, traditional classification often keeps birds and reptiles separate simply for practical study and convenience in the classroom. But it is vital to remember their shared evolutionary roots. This brings us to a crucial question. Why do taxonomists care so much about classifying reptiles based on their skull bones? Understanding reptilian skull classification is like reading the architectural blueprints of vertebrate evolution. The shape of the skull tells us exactly how different groups adapted their jaw muscles to bite harder, chew better, or swallow prey whole. By categorizing these skull types, scientists can track the exact evolutionary pathways that led to the massive diversity of life today, including how mammals evolved from reptile-like ancestors. It also helps conservationists identify and protect genetically unique endangered species. Now that we know why the skull is important, let us look at the specific features scientists examine. The key structures are called temporal fenestrae. These are specific, naturally occurring holes in the skull, located just behind the eye socket. They are not just empty space, they serve a vital purpose. These openings provide extra surface area and room for large, powerful jaw muscles to attach and expand. By simply observing the presence or absence of these holes, taxonomists can turn physical traits into quantifiable numbers, completely removing guesswork and ambiguity from the classification process. To formalize this concept, taxonomists use a straightforward morphological formula. The formula is T equals F sub S plus F sub I. Let us break down what each symbol means. The variable T represents the total number of temporal fenestrae on one side of the reptile's skull. The symbol F sub S represents the superior or upper temporal fenestra. If this upper hole is present, it gets a value of 1. If absent, it is 0. Similarly, F sub I represents the inferior or lower temporal fenestra, also taking a value of 1 or 0. By adding these binary values, we get a clear mathematical representation of the skull type. If T is 0, the skull is solid. If T is 2, the skull has 2 holes. Before we move on, let us see this formula in action with a concrete example. Imagine a paleontologist discovers the skull of a modern tuatara. Upon examination, she notes a distinct opening in the upper part of the skull. Because this superior fenestra is present, we set F sub S equal to 1. She also observes a second distinct opening in the lower part of the skull. Therefore, we set the inferior fenestra, F sub I, equal to 1 as well. We then plug these into our formula, adding 1 plus 1, which gives us a total T value of 2. This means there are 2 temporal holes on each side. Because the total is 2, we confidently classify the tuatara under the subclass Diapsida, which is the hallmark of most modern reptiles. Now, let us apply this systematic approach to a different animal, the green sea turtle. A marine biologist examines the temporal region of this turtle's skull and finds a completely solid wall of bone. There are no openings whatsoever. Because there is no upper hole, the superior fenestra value, F sub S, is exactly 0. Similarly, because there is no lower hole, the inferior fenestra value, F sub I, is also exactly 0. We sum these values, 0 plus 0, yielding a total fenestration value of 0. A skull with 0 temporal openings is known as an anapsid skull. Consequently, the green sea turtle is traditionally classified under the subclass Anapsida, representing the most primitive skull condition among reptiles. To visually reinforce what we just calculated, let us look at this diagram comparing the different skull architectures. On the far left, you see the anapsid skull, where T equals 0. Notice the completely solid bone behind the eye socket, just like we saw in the sea turtle. In the middle is the synapsid skull, where T equals 1, featuring a single lower opening. Finally, on the right, we have the diapsid skull, where T equals 2. You can clearly see the 2 distinct holes highlighted in blue, representing the superior and inferior fenestrae we counted in the tuatara. These structural differences dictate how the jaw muscles attach, fundamentally shaping how each animal eats and survives in its environment. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that reptiles successfully conquered land by evolving waterproof scales and the revolutionary amniotic egg, breaking their dependence on water. Second, we discovered that classifying these animals by their skull architecture is not just a random quirk. It reveals the exact evolutionary blueprints of their jaw muscles and survival strategies. Finally, we used the fenestration formula to objectively categorize these skulls, learning that primitive anapsids have solid skulls, while modern diapsids possess two distinct temporal holes. You now have a solid understanding of reptilian classification. Great job!